both magnet students and homeschool students.
Oh,
So
wow.
so it was a combination.
Yeah.
So, yeah,
Anyway,
yeah.
she's the expert at everything. You want to
Yeah.
ask her?
And was it 10th through 12th grade or?
9th through 12,
It
it
is
was a
9th through 12.
four-year pathway, biomedical science. We used PLTW, Project Lead the Way. I don't know if you're familiar with the
She probably
Project
is.
Lead the Way biomedical science pathway. But we modified it a lot. You know, we pick and choose kind of what was in it. But basically, you know, I have to say that ABE was a thing that really was the foundation for building the program. It was the first program that we brought in to use with our students, and after that, after having implemented labs and the various techniques that we learned, then we just started building upon that to build this four-year pathway. So what I wanted to talk about today mostly is that oftentimes I hear when teachers are given the chance to learn about ABE and implement it in their classroom, the common statement is, oh, I have so much to teach, so much curriculum to cover, I just can't fit it in. And the thing is, ABE really covers the content that, I mean, everybody's got a different program, but it's going to fit into any kind of biology or biomedical science or biotechnology program that you have. And the way that I think is most effective, what research has shown, is to really engage the students in an authentic, real-world application of science. Students don't respond as well, do not become as engaged when you say, okay, we are going to cover biology, and first we're going to do chemistry of life, and then we're going to move into central dogma, and then we're going to do genetics, and then we're going to do physiology or whatever, because that's the way our book has laid things out, or our web resources are laid out. It's much more effective if you can have some type of real, authentic project that the kids are working on that brings out all of this information and content. And storyline type curricula do this. ABE is a storyline type curriculum in that basically you're showing what biotechnology can do in order to improve the health of individuals, particularly those students who have type 1, or, you know, people that have type 1 diabetes. So, you know, there is a real-world application to it. Students see this and they're more engaged. So, when I try to bring in any kind of program or research or you know, like technology into the classroom, I always want it to fit in to that type of situation, that there's a storyline that, you know, it's relevant to the students. And the one thing I try to do is I try to let the students raise the questions that they have, and what do they need to know in order to, you know, solve the problem or answer the question. And I'll give you a related example a little bit later, but you know, that's what I do with ABE. And the driving question that we use this is our freshman biology class. All of our freshmen do the ABE. And the question is, how can biotechnology improve, you know, human health? And that's the driving question. And, you know, basically it's like, what are these pharmaceutical companies doing that is going to help, you know, improve human health? And they come up with the questions. Basically, your foundational concepts are built into the the storyline where the students ask the questions and then the content comes out. And the one thing that's very important about that is when you do bring the content in, you do just enough, just in time. You don't say, okay, we're going to do this, ABE program, and you're going to need to understand the central dogma. So then you move into that stand and deliver kind of thing, saying, this is what the central dogma is, and you go into every detail, and by then the students are turned off. You give them just enough, just in time, of the content so that they can move forward in the project or in answering the question. The other thing is you want to try to make it as much like real science as possible. So you try to create a situation in your classroom that mimics, say, a lab group, that's conducting scientific research. And that involves a community of individuals that are asking questions, making decisions about how to move forward, prioritizing the questions, and so on. And that's what develops real critical thinking that is going to make them valuable members of the scientific community. Another very huge component of science is models. When scientists do research and they're trying to, discover something and answer questions, as they move through their research, they create models to explain it. So it's important to have the students trying to develop their own models as well. We're all familiar with that. We talk about with enzymes, the lock and key model, or signaling pathways where you have, a ligand that binds to a receptor, which causes a conformational change, which then, produces some type of response within the cell. So models are very important, and getting students to learn how to do that is important, because that's how science works. That's how scientific knowledge is increased. So basically, you try to develop that in the classroom. And having built upon ABE, we've done that with a number of very authentic programs that we've done a lot of times articulating with community college, universities, and so on. So just to give some examples, and if you want to know about these later on, I can go into more detail, but we do DNA barcoding. Are you familiar with what DNA barcoding is? Okay, so basically, DNA barcoding is where you can identify a specific species of an organism based on a particular sequence of DNA that's unique to that particular species. So, to give you an example, if you take animals, vertebrates, animals, the DNA barcode that is able to help you identify the species of an animal, it's the cytochrome oxidase subunit 1. So in oxidative phosphorylation, you got the electron transport chain, and the very terminal acceptor of the electrons is cytochrome C oxidase, and that's the enzyme that takes oxygen from respiration and combines it with the electrons that are transferred down the chain, along with the hydrogen ions that had been pumped across the mitochondrial inner membrane to produce water. So basically, that's the terminal step in what happens with oxygen, why we breathe. Okay, that particular enzyme has 13 subunits, and basically subunit number 1 is a gene that is encoded in the mitochondrion. And a sequence within that gene is what's used to identify all animals, okay? And what happens is it's got enough similarity, but by species, there's differences. So if there's a 2%, basically the cutoff is a 2% difference. If you've got less than 2% difference, then it's the same species. And that's what identifies a species. So, you know when you go to the market or the store and they have barcodes that identify what you're purchasing? That sequence of ATGC is what identifies a species. The mitochondrial cytochrome oxidase subunit 1 barcode, a sequence within that gene. So basically, in order to do barcoding, you just basically amplify that sequence, you sequence it, and then using bioinformatics, you can identify the species. So that's what our students do. So, I mean, you could learn about that and do it. You can get lab kits and so on. But then you're just kind of going through the motions. So we work with CSUCY, Cal State University, Channel Islands. I don't know why they named their college CSUCY, but anyway, we go out to Santa Rosa Island twice a year for a week, 1 week in the fall, 1 week in the spring. We collect indicator species. They're mostly pollinators, insects. We barcode them, and what we're doing is we're monitoring the biodiversity for CSUCY while they do restoration on Santa Rosa Island. They're trying to get it back to its, more natural, endemic type of ecosystem. But we're the ones that monitor the biodiversity so they can follow what's going on with their project. So, in addition, we've got coastal marine biolabs that have 2 very amazing curricula. One's Neuro Lab, the other's Toxin Lab. These are storyline curricula that's developed, and it basically gives, phenomenon. I'll just do Neuro Lab as an example. Okay, in Neuro Lab, the students see video of this condition in people where they go to do a task with one hand and their other hand mimics what they're doing. So, to make a long story short, basically what happens, it's a genetic disorder that has caused the neural path finding that occurs during development to go awry because of mutations in the secretion of a ligand and a receptor, so that normally as the spinal neurons develop, they cross over, decuss, and what happens is in these people that neural path finding is mutated, altered, and some of the neurons go straight down. So instead of the right brain always telling the left side what to do and vice versa, you've got the right brain telling both sides what to do, and so you get that mimicking. It's CMM, it's congenital mirror motor disorder.
Yeah, I'm a PhD physiologist and I've never heard of that. So
Okay, well, yeah, anyway, it could have been anything, but the point is, they take that phenomenon of a student seeing what's going on. Why are they doing the same thing with their other hand? They develop questions, and it takes it all the way through from identifying, is it a neurological or a muscular disorder by various imaging technology, what genes are involved, what do these genes do, and there's evidence as it goes along. So they have to use the evidence to create a model that explains CMM. So that's...
So it's a receptor ligand during the very early development.
Oh, yes,
right?
yes,
So during
yeah.
the neural development.
Yeah. So, the thing is, again, this could have been any phenomenon that you want to bring in. The point is, it teaches a ton of biology, okay? And the kids aren't sitting there, okay, now you have to learn about this topic. And it's like, okay, whatever you say, I'll do, I'll do it because I want to get that A. It makes it engaging for the students. They want to solve the problem, and they're doing it the way that science actually happens. Toxin lab, just as another type of example, people create they have shellfish, they make a soup, and people get sick and they go, they have to go to the hospital, and they get like amnesiac kind of thing. They also look at some of the sea lions and stuff. There's marine mammals that start going crazy and stuff. And again, it's a whole thing of what's causing this, what kind of disorder is it, what's the basically this is one where it affects the mitochondrion, which secretes chemicals, which then have an apoptosis effect, and then it's destroying cells that are involved in, basically neurons and so on that are involved in the brain, and that's why they are amnesiac. I mean, after a while it can go away.
That's a strong theory with Alzheimer's.
Yeah,
So
yeah.
the mitochondrial.
Yeah, yeah. So anyway, we also have, there's Ellison Institute of Transformative Medicine. It's a cancer research institute in Santa Monica, and what we do is they create a cancer phenomenon where our students learn how to look at cancer, figure out what type of mechanism is involved in it. They get data along the way, and then they learn about personalized medicine, and they have to decide, should we do this chemotherapy or should we do some type of gene editing. So again, it puts them in the position as if they're real scientists, and it's an actual phenomenon. And then we have our identity project. We're a gold certified linked learning school. Are you guys familiar with linked learning? Okay, so that's a thing where we have a pathway, and it is a linked learning pathway. So it means that our students in the program are sort of cohorted in not only their science classes, but math and English and social studies, the fluff stuff. Anyway, so that they have each year, each grade level, they have 2 interdisciplinary projects they do, and one of them is, and this is in the 10th grade year, we have the identity project. So, like for example, in biology, they learn a lot about what determines their identity. So there's genetics, but there's also, environmental influences and things like that. One of the social studies teacher teaches AP Psych, so she goes into things about, gender and identity and that kind of thing. They also do things, because they're taking the social studies for our program is world history. So they start kind of trying to create a family tree, based on their ancestry. But the cool part is in their pathway course, they actually, each student gets their own DNA, and we isolate this is again is a mitochondrial region. It's the control region, so it's not a genetic. So one of the things is when you do students' own DNA, you can't do anything that relates to like their health or their anything like that. Well, the mitochondrial control region doesn't. Okay, it's just like a regulatory thing in the mitochondrion. So anyway, they PCR that, and they sequence it, we actually have Sanger sequencing, and we also have whole genome sequencing that we've introduced and we've got the equipment for. But anyway, they sequence their own mitochondrial DNA, and then what they do is the out of Africa theory, where they look at the different haplotypes that originated, and so they can see with the out of Africa, were they in the haplotype that went to Europe or to Asia or to Australia or whatever. And a lot of times then they can connect what they know about their family tree and actually figure out how they went from out of Africa all the way through time to Reseda High School. So, I mean, it's kind of a cool thing, and they,
Reseda.
yeah, I mean, because that's where they're in high school. But so that's another, basically it's again, it's not, and the reason I like linked learning is because the kids learn, oh, you don't do math just in math class, and you don't do English just in English class, and you don't do social studies just in your fluff class, and you don't, and so on. So it basically teaches them that you need all of these classes to solve real world problems, and so that's what I like about it. So those are just some of the things that we've been able to build upon ABE as our baseline beginning program over the years to add on and try to mimic the types of things that we do here in ABE. Okay, so now I'm going to get to some things you can add in because you want to have the kids learn a lot of the skills. You want to take advantage of ABE and all that it has to offer. So since we are linked learning and our kids have to take art, one of the things that our art teacher does, besides teach medical illustration as one of the units in their art class, we do a pointillism activity. And I have no art background, but so basically pointillism is a style of art from basically it's post-impressionism, and so you can actually train the kids to really get good at micropipetting by having them do a pointillism activity where they create, and there's actually you can get a kit from Edvotech, or you can just do it on your own. I put in the Edvotech thing. But basically, you use dyes and the micropipettes, and they basically make art, but it's just points. So that was a shark, whatever. So anyway, and then, there's like a more advanced example of pointillism. The ones that the kids do aren't quite that advanced, but the other thing is they have to pipette a lot to do something like that. So you're really getting, I don't know if you find that the kids don't have the manual dexterity that they just don't do, yeah, they're really good at, texting, but like doing anything else, yeah, not so much. Like they haven't done, well, I guess like in kindergarten, their scissor skills or whatever. Anyway, they don't do crafts as much or that kind of thing. But that's a real cool thing. And then there's the dog drool and the kitten paternity labs. Okay, so for our pathway, we have a summer bridge. It's a 3-week, our incoming freshmen come, and what we're doing is we're training them, we're trying to teach them about how to do like project-based learning. So a lot of them have never done that. A lot of times they're coming from different schools, so they don't know each other. So we're, getting them to be collaborative. It's sort of a thing that we're trying to prepare them for our pathway. And doing the dog drool and kitten paternity labs, what those are basically is they learn how to pour gels and run a gel and do micropipetting. And, it just gets them so that during the class, during courses when they start as freshmen, they're not learning those skills at that time. They've already developed them. So those are great, activities you can, if you have middle schools that you want that are feeder schools, articulate with them and get the kids so they know how to do pipetting. You know, we do a lot of things where we have Saturday science days or STEM workshops where we bring the middle schools and the elementary schools in, like on the weekend. We just did one yesterday at Chatsworth High. And what it is it's kind of the idea of a farm system in sports where you're getting your, the up and coming, future stars for your high school. You're getting them, trained and getting them to become part of the team and that kind of thing. So yeah, it's been a great thing for our program. Okay, so I want to specifically talk about how I use ABE with our ninth graders. So again, it's not like, oh, we're going to do this ABE program and, this is what you're going to do. It's introduced as part of a driving question of how does like biology or biotechnology improve the quality of life, because if you're going to go into something like science, hopefully you have a reason for doing the work. So obviously it incorporates things that you have to teach anyway, the central dogma about enzymes and so on. But basically, we introduce it as, the way they do. It's like the question about diabetes. How do you help people with type 1 diabetes? What's the problem in type 1 diabetes? Oh, they can't make insulin. There's some type of mutation. And so they have to have insulin. And, historically they look up, oh, we used to get it from farm animals and stuff like that. Sometimes there's problems. Wouldn't it be great if they could have human insulin? Okay, well, let's see how you do that. Okay, so basically we take them through the whole process. Okay, so now you've just experienced how biotechnology works. What other kinds of applications are there? Biotechnological applications are there. So the kids research all kinds of things. The CRISPR, in vitro fertilization. I mean, it's like there's tons of. I mean, they can look at agriculture, GMOs, things like that. What kind of technologies are out there that are, where they're trying to help the society? And there's like, environmental. I mean, whatever. I mean, you can even skew it to whatever. Like if you say you're an environmental magnet or you have an environmental program, you can look at, how are they, getting bacteria to, possibly produce nitrogen for farming or, clean up oil spills and things like that. So you can, the thing is the fun part is like figuring out ways to relate it to whatever your program is. So, our students, we're a biomedical science pathway, so basically they research all of these different types of biotechnology, and then in the fluff classes, they actually learn rhetoric and how to do a debate and all of this stuff, and then they pick, a topic is picked, a debate topic is picked, and the kids debate, is this ethical? So they learn about ethics, and then they debate, is this ethical or not? So that's how we, make it interdisciplinary and see how it's combined. And even like bringing the math in, having the kids, have to use like statistics or, like percentages and stuff in their argument. You know, they have to have mathematical components. Now, one of the things is, you know, trying to make it real world in addition to debating it. Once they've completed this and they've looked at, you know, how a particular product can be made from a particular gene, they have to research what, if you were going to be a company, or if you worked at Amgen, what protein product would you want to make, and why would that be the best one to choose? And so they write a proposal of, you know, oh, I think we should make a clotting protein for hemophiliacs or whatever. But, you know, they have to look it up and research it and try to justify why that should be the best, you know, protein or the best gene product to produce. And one of the things we try to do as often as possible is have what we call high stakes assessment, which means we bring in industry professionals, so the kids have to present their research and their, you know, projects and things to, you know, experts, because God knows they don't care what I think. So, like for this project here, we get people from UCLA Health, and basically they serve as a judging panel, and the kids, you know, get up and do a presentation and present their evidence for which protein, you know, which gene product should be produced and why. And, you know, they're judged by industry professionals. We've even had, one year we had two people from Amgen come and judge. So, you know, it's like when I used to teach physics, I would have the students design a roller coaster, do a technical report, that kind of thing. And they had to present it to the engineers at Magic Mountain. And, you know, so they felt that it was authentic, real world. It wasn't me giving them an assignment to keep them busy. And that really helps with student engagement. One thing is we all have to do our differentiated learning for our various students. And, you know, so basically, again, you can go into this at various depths with, you know, different students by asking them, you know, certain students specific questions or having them research specific things. The other thing is if you have some really, you know, enthusiastic and bright students, I occasionally let them do the colony PCR. Are they trained in the colony PCR here, or is that?
This is the abridged, so we're
Yeah.
skipping that.
Okay. Well, anyway,
They can come back.
yeah, there is an extension that adds in the colony PCR to verify that you've cloned the gene you think you're cloning and so on. But then in the AP biology class, that's done, again, we repeat the ABE sequence in AP Bio and add the colony PCR. You know, I think oftentimes when you go through something the first time, you don't get it all. So doing it a second time and then making it at a more rigorous level really strengthens what you've already built as a foundation, and then expands what they do, you know, at the next level. So those are some of the ways that I use the ABE. Okay, now I'm going to give you, this is my example of how you can take and do a project, do project-based learning, and not say, okay, we're going to learn genetics and some other biotechnology, you know, like where it's, I mean, like genetics. I don't say we're going to learn genetics, just like I say we're now going to do the chemistry of life section. I just have the kids see that plastics are really bad. They're all in the ocean, they're in the air, the microplastics and everything. What are we gonna do? And so my freshmen actually make a biopolymer as a bioplastic. And you can do that by covering chemistry of life. This is how I cover genetics, and I don't say, we're doing genetics now. It's just part of a project, a real world project. And so we have just talked about with ABE, how do you use like molecular biology to improve, the life of people with diabetes. Well, what about, there's a lot of genetic disorders. And so I use the Exploring Genetics through Genetics Disorder from the University of Utah. It's the Learn Genetics, Teach Genetics. I don't know if you've seen that before. So in that program, they have already set up 5 genetic disorders. Each of those disorders has like 5 or 6 mutations, alleles, mutations in the allele that affects, that particular, that causes that disorder. And so over here on the side, this is an example. You're given the healthy sequence and then the mutated sequence, and like for example, that's a point mutation, okay? But within that program, there's point mutations, there's indels, there's insertions, deletions. They also have transpositions and inversions. So they get to learn about all the different kinds of mutations. The class is broken into groups, and each group gets one of the disorders. And I've even, if there's somebody who I ask them, what are some of the genetic disorders you know of? So if they're really interested in getting a different genetic disorder, it's not that hard to add one in and do the same kind of thing. You just, you can research and use bioinformatics to see what gene is involved and get mutations. You can get them from bioinformatics, from NCBI. So, I've done that with other disorders if they have a particular interest. But in any event, what they do is they look at a mutation, and what does this mutation do? So they have to see, does it change the amino acid? Does it introduce a stop codon early on in the gene product or at the very end? And then how does that affect the protein that's made? And then what does that protein do? What's its normal function? And now what's getting screwed up? And how does that affect the individual at the cellular level, the tissue level, the organ level, and so on? And it's all, that's all laid out in the teaching genetics through genetic disorders. So then they also have to do a pedigree to analyze a pedigree to determine what type of inheritance is involved. Dominant, recessive, maternal, that kind of thing. And they've got all examples in there. So they learn the Mendelian genetics, but that's kind of getting old hat now because molecular genetics is really where we're at. But in any event, it does cover it. And then what I stick on to the end of it is I teach them about CRISPR. I give them the article, I don't know, well now I think Europe just approved the CRISPR gene editing to treat sickle cell disease. But I give them an article that, when the research came out about how it was done, and before the clinical trials, but you can follow up and say, in Europe it's now approved and it's, going through FDA approval here. But basically, I have them figure out what they have to do to use CRISPR gene editing to treat their genetic disorder. So, they learn about CRISPR, and then they learn about CRISPR gene editing and what they have to do to fix the gene, and then there's a cool website. It's a bioinformatic website. It's called the Prime Design, and they put in. I know it's small there, but you put in how you want to change. You're going to create your guide RNA that drives the CRISPR system. You're going to change your guide RNA to make the specific change, whether it's a base substitution or an insertion or a deletion, that kind of thing. You plug it into the bioinformatic platform, and it gives you, probably can't see it down there, it gives you the guide RNA that you would synthesize, and now they can treat their patient that has whatever disorder of the allele they have. So, never did I go in and say, now we're going to learn genetics, or now we're going to, it was, how can we help people with genetic disorders? And so they think they're, actually solving a problem. But you're really covering the curriculum that, you have to teach, or you want to teach.
Is that what you mean by the problem-based education?
Uh-huh. Yeah, it's a problem. It's not a chapter in the textbook. Okay, that's the big difference. Okay, so there's two things I want to talk about. I don't know, are they going to cover? Okay.
We mentioned it, but please don't override.
Okay, so you're getting the ABE program, so you can take that back to your classroom. You can take additional trainings to expand that, but they also provide amazing opportunities. I'm assuming that you're gonna cover the use of lab exchange.
Yeah, oh
Yeah.
yes.
Okay. Well.
They think I work for Lab Exchange by now.
Okay. Yeah.
I'm
So
getting a commission.
that's got a ton of resources on there. But what I wanted to talk about is the ABE volunteer program. And we are so lucky because we are so close to Thousand Oaks and, well, I guess it's Newbury Park. I don't know. Anyway, it's
Both.
Amgen.
Both.
Amgen.
Also.
Because you can get these amazing people from Amgen to come to your classroom and let the kids know about what their career is like. And we have been so fortunate that and it's not just they're going to get some lab scientist to come in to tell you how they, do the same thing that ABE does where they're creating a therapeutic, okay? We've had the president of the Amgen Foundation. So you've got somebody involved in, funding, different projects and stuff. We've had the vice president, or I think she's the vice president of external research development. That's where Amgen, is looking at all of these startups and companies, and when they see something that a collaboration would be, just totally beneficial, they, bring that in. We had a lead for a project lead for a cardiac therapeutic. The coolest thing, because maybe a lot of your kids, they, especially the freshmen, come, what are you going to be when you grow up? What are you interested in? I'm going to be a doctor. They all say they want to be a doctor. Okay. One of the best things that this particular guy talked about was his parents. Because the other thing they do is they talk about their journey to their career. And one of the things that this one presenter talked about was how his parents wanted him to be a doctor. And instead he went into biomedical research and, worked for Amgen to create a heart therapeutic, a heart drug, okay? And when his drug reached clinical approval and got FDA approval, he told his parents, this is why I went into biomedical research. In the amount of time I've been working, I could have seen X number of patients and helped them. by going into research and creating this therapeutic agent, I can help 100 times more people. And that's why I went into it. And I mean, that really resounded with the kids. Just like when we had the president of the foundation talk about his job. You know, it allowed some of those students maybe that weren't so much the sciencey type, but you know, there are other careers that you know, can help you you know, make a difference in the world and use your background knowledge of science, but you don't have to be a scientist. We also got one of the Amgen volunteers who was in charge of clinical trials for a drug. And this person happened to be an epidemiologist. And since we're a CTE pathway, we have to have a CTSO, a career technical student organization, which for us is HOSA. Are you guys familiar with HOSA? It's future medical professional. So it's anything, even allied health, and it's an organization, it's international, and kids study things and they compete at the state level and they can go to the international competition and all this stuff. So it's like a, you know, it's like a science club for people that are interested in the medical profession. So anyway, because of her background in epidemiology, she helped our students who were doing the epidemiology competition with HOSA, and also the kids learned what it takes to get a pharmaceutical through the clinical approval, the clinical trials and approval, which most of them don't even understand. I mean, you know, not only students, but the public doesn't understand what it takes to get through. And the other thing it does is it helps people understand that when the FDA has approved something, what, you know, how confident they are in the efficacy of that therapeutic, or, you know, sometimes devices or whatever. So they've got a whole toolkit that helps you prepare and get this volunteer program to send somebody to your school. The kids are just really inspired and just really eager to interact with the volunteers from Amgen. The last thing I want to talk about is after you've done ABE for a while and you know, that kind of thing, they have this thing, Master Teacher Fellow. It's an opportunity that they provide for a teacher who wants to investigate something that they're interested in and create a curriculum. And I was able to do this 2 years ago, and the thing in which I was interested was synthetic biology. And I was, you know, just enthralled when I learned about this. I you know, took like an online kind of course on it. And they really say this is going to be what they call a disruptive technology. Disruptive in it's going to blow everything, you know, it's like the computer, the laptop, things like that, just completely change things. So that's what they think synthetic biology is going to be. So it's really, it's a combination of biology and engineering and computer science, because it involves all of those. And so basically, I created a curriculum. It resulted in us setting up an articulation with a professor at USC who does synthetic biology. And so what we did was we created a sort of a articulation between his lab group of grad students and postdocs and the professor with our school. And during COVID, we did it virtually, but then the next year, when we didn't have to play COVID, you know, the professor would actually come out to the classroom, and basically his lab, they're creating organoids. And what that involves is you create genetic circuits. So these are DNA sequences that are synthesized, you know, and you put them into plasmids, and then you put them into tissue culture, into cells, and they're going to do what you've programmed them to do. And so we did some synthetic biology labs, bio builders, yeah, they have them. So there's a few that Carolina Biological Cells. Anyway, so the kids did some labs, and we created, basically the professor, you know, taught them how to do these genetic circuits, and showed them examples of things that they had done in his lab. And then he gave, there were like 5 or 6 challenges to the kids. And the kids worked on them, and then we got to go to USC to his lab, and the students presented their solutions to his lab group. And I mean, you know, I mean, if you want to make a kid feel like I can do this and I can be successful, and so on, that's a kind of experience you give them, and you know, they walk away, you know, with a passion for what they've learned, and knowing and having the confidence that they can pursue this, you know, in their future. So, you know, basically, they wanted to create different organizations of particular cells to get them to, you know, have some kind of tissue, cell morphology with certain cells on the outside, certain cells on the inside. You know how kind of in development you have the ectoderm and the mesoderm and the endoderm. So you're creating different locations for different cells. There was one challenge that involves cell signaling, getting cells to do things based on when other cells had a certain stimulation, you know, like you add a ligand and so on. Anyway, so our kids, you know, worked through it and presented to the lab group at USC, and you know, that, I mean, you basically put them in a real world authentic situation. So that was what came from that opportunity that Amgen, ABE, provided for me to be able to develop. So, yeah. So that's kind of what I wanted to share. So,
Thank you
mhm.
for sharing.
Yes.
Any questions? So, yeah, thank you for mentioning.
Have you seen
the
success
vascular
with students going on into those various?
Oh, yes.
Oh, yeah. Yeah. The thing is, if you can give your kids these opportunities that others do, I mean, you know, basically to get into some of the more competitive programs, like for example, you know, like the Tier 1 colleges, all the kids that apply, they all have straight A's, and you know, they've all done some things. But if you've got a kid that's done something like this, or I mean, our kids even interned for Karen to help do the Amgen ABE kits, you know. But having done like these types of experiences, these real world authentic experiences, you know, like the DNA barcoding, they presented at NASA JPL's regional, we got to the regional science fair or competition, whatever they called it, you know, and it's like they won it hands down, and there were students coming up to their presentation poster and saying, wow, we just learned in AP Bio about, you know, how you create a phylogenetic tree from DNA sequences, and your students are doing it. You know, I mean, stuff like that. That's what gets them into, you know, the prestigious institutions. But the one thing I want to say is that we get a lot of kids that go right here to Pierce, and they do, because LAUSD, you get 2 years of free college, okay,
Really?
in the community,
Yeah.
with the community colleges.
No kidding.
So, and, I mean, I
For
just,
being an LA resident? You
the
get-
LAUSD,
Oh,
yeah,
LAUSD.
all the kids in
So
the
if you're from
district.
LAUSD, you automatically get 2 years?
2 years of free college, plus, like, I've got 2 kids right now, well, I retired in June, but I've got, no, actually 3 kids right now that are over there in the, taking Pierce College. You get to take Pierce College courses for free
More
while
time
you're,
too.
and, I mean, yeah, all the community colleges,
Right.
yeah.
So essentially, it becomes a feeder program
Yeah.
for the college.
And
Yeah.
now they're starting
community
with
colleges.
bachelors at some of these.
So-
Yeah,
Moore
like
Park
Pierce
years.
and,
Yes.
Pierce, Moore
And so
Park,
now they
and,
can get
and
bachelor's in
Mission.
biotech.
Yeah, we
Yeah.
heard about the Moore Park one.
Yeah,
Yes.
You guys
Yeah.
talked
Yeah,
to-
I'll tell you, I'll give you a list today when I do my presentation. I'll give you a list of with the location of these colleges and whether they offer AS or BS.
I think one of my
Yeah.
friends is a teacher at your school, Mary Ann Mackey.
yes.
Yeah.
Yeah.
So,
Mackie's retiring this year. We had a race, I beat her.
oh,
Yeah,
so
no,
she's
I've
amazing.
known Mary Ann since I started at Reseda.
Yeah.
Yeah, yeah. So
But,
do you do Envirothon or?
Yeah.
Yeah, okay. So you could, you could take the environmental science
Mhm.
approach with a lot of the curriculum if you want to develop those kinds of things. Where do you teach?
Granada Hills Charter High School.
Oh, okay, yeah.
Yeah, we'll show you can do
Yeah, and
bioremediation.
I teach Enviro and Bio, so,
Yeah,
yes, it's nice.
yeah. Yeah, I'm more on the cell and molecular type.
Too.
Yeah. Yeah.
One time I taught for National University and they, for microbiome, and they did a project, it was very interesting, where they'd take dirt, okay,
Oh,
then they'd have to,
eDNA?
ooze, I'm not sure what, well, anyway, they would look at various bacteria.
Okay.
from
The
dirt and
microbiome.
then-
Yeah.
And see if it was, what do they call it, predator bacteria.
Okay,
Which literally,
yeah.
you'd see on the thing, a little
it-
circle where no other bacteria were.
Because
except
they were
that
eating them up.
Yeah,
Yeah.
yeah, but that was the start of how antibiotics get going.
Yeah.
Is that, bacteria, or even mold, well,
Yeah.
they weren't looking at molds actually, this
Yeah.
is bacteria, but, that beat up other bacteria.
Yeah. Well, I
And
actually,
it was a collaborative project, that was the other thing, it was part of, I think it was Princeton, or, some back east, had all these different, so it was like
Yeah.
a think tank, or a, what do they call those, anyway, collaborative project.
yeah, I actually had 2 students, this, only high school kids would do this. I had 2 students that worked with a professor at UCLA on, who worked with,
I went there.
yeah, I taught there from, let's see, well, anyway, back in the day. But I had 2 students that worked with a teacher there doing microbiome stuff. Their project that they did was, do people with acne compared to people with clear skin have a different microbiome? And is it that the people with acne, that the unique bacteria are causing it, or the people with clear skin, the unique bacteria preventing it? And, basically, like in science, it was kind of a little of both. So, yeah, but, yeah. And that's one thing that there's 2 things I would recommend, reach out to your local community colleges and the colleges and universities. Like I had even some of those kids that are over there now, I had about 8 or 10 students that did a project at CSUN where they did gene annotation. So they basically annotated Drosophila. It wasn't Drosophila melanogaster, it was other species of Drosophila. They used bioinformatics, and bioinformatics is becoming huge. Get your kids to do at least a little computer programming and stuff. But over the summer with CSUN, they did gene annotation where they all annotated different genes, and it's published. It goes on to NCBI. But connect with your community colleges and colleges and universities. I mean, like I said, we articulate with CSUCY, Pierce, Mission, CSUN, UCLA, we do coral proteomics, that's a bioinformatics thing,
Oh.
where the kids, they analyze LC mass spec, peptides to figure out the sequence of a protein for corals and also fish otoliths. But it's basically they're looking with the coral, what proteins help them survive, like climate change. And so it's just basically UCLA does the LC mass spec, our kids analyze the data. And, those kinds of collaborations are, if your kids can do those, again, that's that step up that they're going to have over other kids. The second thing is, how many of you have a CTE credential?
Oh, okay.
All right.
Great. And-
So the
In what?
CTE
Advan-
credential is one of the biggest things that help my program grow, because that's where the money is.
Yeah.
Okay, so I don't know, what's your typical, just regular science budget for the year? How much money do you get? Ballpark figure,
0.
okay? I'm not surprised.
We just, I was just telling him yesterday that the superintendent just removed all our funding for the pilot school.
Oh, wow.
Yeah,
He doesn't do pilots, he supports other type of setups, so.
yeah. Well, if you're in CTE, okay, so
What's
this
that
is,
stand
this
for?
is why, career technical ed. It's like,
Okay.
they used to call it,
I tried to get one for
Vocational.
forestry and natural resources.
Vocational, yeah.
Oh,
it was,
Votex.
what was
Right,
it?
Yeah, yeah.
Vocational.
Yeah, I mean, it's that
Votex.
kind of thing.
So,
So,
yeah, so at our so I teach community college level, and
Yeah.
most, they're in small systems, and they often have a VOTEC school of some sort in that system, right?
yeah,
So that-
yeah,
And I think they have used that
Yeah.
that CTE.
Our
Yeah.
CTEs are now, all that vocational ed is all pushed out into our public schools now.
Yeah.
Yeah.
At high school level.
And
Well,
our
and
CTE
middle-
There
biotech
is no,
person
like, kids don't
just
go anywhere.
left and went
They-
to
They
Montana.
do, right there,
So
So we
interesting.
don't have a
yeah.
CTE
So John, are you
biotech
credentialed
Oh,
in
you're
biotech
kidding.
or in
Yeah.
medical?
but you
Right.
could
Yeah.
get a CTE credential, because, I mean, so, I
So like
mean,
MAs and all that.
okay, usually CTE teachers come from industry.
Yeah.
But you can use the relevant courses when you were in college. So like if you want a biotech one, you can use biochemistry, biology. So like I have two of them. I have in the health science and medical technology, and then I have engineering and design, because I used to do robotics and engineering for a while. Anyway, but you can use your college courses. If you do anything outside of, your regular daily teaching activities, like if you have a science club, or if you do things in the summer, or anything like that, or if you've done research, like if you got your master's and you did some research, that counts as industry hours, and then you can get your CTE credential. And I get over the past, I don't know, 6-8 years, I've been getting anywhere from $50,000 to over $200,000 a year for stuff.
That's what I'm
And-
trying to do for forestry and natural resources, because I do forestry challenge and envirothon, but I also have a master's in con bios.
Yeah, so that's the thing, that's where the money is.
And they're looking for CTE
Yeah.
credentialed teachers.
It's just so
as a-
hard to,
yeah.
like, I looked at it through Ventura County Vet, it's just hard to get those, how do you document all the hours? That it's, their
Okay.
worksheet is really difficult.
So basically, you need your transcript if you're gonna use, the, courses, that relate to whatever field you wanna, get a CTE credential with, okay? And I'll just tell you, if you're really old like me, I contacted UCLA. I was at UCLA, I spanned 3 decades, the 60s, 70s, and 80s. Anyway, I had to get my transcript, which started back in the 60s, and I called UCLA and they said, oh, just use your email address. And I said, well, I don't have an email address. They didn't have it then. And then they said, well, what about your registration number? Well, I haven't kept stuff. That's been so long ago. And they said, well, can you go online and put your name in and then they'll find your email address? And I said, when I went there, there was no internet. And there was like this dead silence. It's like the student worker that's, you know, doing this little job. It was like, how could that ever have been? So, yeah. Anyway, you need your transcript. If you did research, like I just, you know, wrote to the, my major professor and got a letter. I mean, that's all you need is a little letter.
So I
And-
could get one from Cal
Yeah.
State Long Beach and from Forestry Challenge.
Exactly,
Oh.
exactly.
I didn't realize
And if you did like stuff in the summer, if you did like Sierra Club stuff or this, that, or the other, you can, all you need is letters. So like for engineering, when I did robotics, you know, I just got a letter saying, well, I you know, got trained to do CNC, so I spent like 3 months doing that, and then I'd done it, the robotics for a number of years, and you know, I got people to write letters about the time that I'd spent, and that, you know, so the only thing I had to do was to take, because I already had a regular credential, was to take one course. I had to take one course, and I think it was like the one of whatever they call it, like the FABAL, like working with different ethnicities or something like
Oh,
that,
okay.
you know.
Huh.
And I think you have to get first aid certified, but you know, most of us do that anyway, because, yeah. But that's
Thank
where the
you.
money
Yeah, 'cause
is.
that's where I mean,
Yeah.
I could easily bring biotech into AP, Enviro, and all my classes like that. It's not
Yeah.
difficult.
Yeah,
And I do
you
a
can
whole
stay
corals
inside.
unit in bio.
Yeah,
That'd
yeah,
be nice to do something with it, biotech.
You need
Yeah.
$3,000.
Thank you, Wendy.
yeah.
Thank you so much. Do you want to stay with us? We have lunch here already.
Super easy.
So,
Oh,
because,
sure,
yeah,
sure.
why don't you join us for lunch? So if you have more questions for Wendy, she's gonna join us for lunch. So, and